Yeah, it's my pleasure to welcome Dr. Eun Ji Chung here from University of Southern California. Um, I won't go into too much detail because I'm sure she'll tell us all about what she's been working on and what the exciting things she's found. But she came, I guess, did her undergrad at Scripps College, then went to Northwestern, and then did a postdoc at University of Chicago, where she got a very prestigious K99 award that then transitioned her to USC. And while she's been there, she's gotten, I don't know, too many awards to read off, and some very nice funding, NIH Innovator Awards and things like this, which I guess seeded all of the great stuff that she's here to tell us about. So without taking any more time, I'll let Eun Ji take it away.
Yeah, thank you so much, Ben and June, for hosting and that introduction. I'll go ahead and share my slides. All right. Guys seeing the regular view? Okay, perfect. Yeah, so as Ben mentioned, I'm at USC and I've been there now almost 6 years, and I'm in the Department of Biomedical Engineering. And I'm also affiliated with a couple of different departments as well, just because our work is, very closely tied with clinical applications and developing different strategies and solutions for places where there could be better solutions. And primarily our work has focused on nanomedicine. So I'll talk a little bit about that, and give a couple of different stories that have been going on in my lab since I arrived. Okay, so yeah, just as a brief overview again, are you guys seeing my pointer, by the way? Do you see this or? Okay, great. Yeah, we are a more broadly biomaterials and nanomedicine research group. So that just means that we can design synthetic molecules. We can also make use of natural polymers like collagen, and then ultimately develop some sort of assembly or architecture that's utilized for an application of interest. So this picture kind of depicts our design, motivation, and kind of reiteration process. And just to get everybody on the same scale and thinking about nanomedicine and the nanometer scale, I like to kind of show this where you can take a piece of your own hair, and we know that that's about 100 micrometers or about 100,000 nanometers. And if you go down a fold, you've got red blood cells on the order of about 10 micrometers or about 10,000 nanometers. And nanoparticles, of course, they can be anywhere on the nanoscale, but they typically hover around 10 to 100, maybe up to about 500 nanometers. So I mentioned that nanoparticles are small, but when you compare them to small molecule drugs like chemotherapeutics, they're actually quite big. So small molecule drug, let's say they're about a nanometer or less, and a particle is about 10 to 100 nanometers. And so that difference between a drug and a particle is sort of similar to a soccer ball compared to a Goodyear blimp. And so you can kind of imagine how many soccer balls can fit into a blimp and be carried, right, to a specific location. And so that's really the analogy here. So again, nanoparticles are large when we think about them in comparison to drugs. And so because of that, they can actually fit multiple drugs and be used as a carrier. Sometimes I think about them even as a courier system where they can go to the right address of interest. And because of some of their physical chemical properties and their size, they don't necessarily always access not all of the parts of the body. And so I'll kind of touch upon that as a theme today, how you can design particles based on their physical chemical properties, as well as targeting and biochemical properties for I guess, more of a targeted double targeting approach. So nanomedicine, though, has been around for a while. I think this is sort of one of the first success stories, which is doxorubicin in a liposome called Doxil. And so this is already approved. It was approved in the 90s, and it's used for a variety of different types of cancers. And I guess now, kind of fast forward to modern day, modern times in the last 2-3 years, we know that nanomedicine has taken sort of a huge, it's just exploded, right, with the COVID vaccines, where we have mRNA that encodes for a piece of the spike protein that's encapsulated within a lipid nanoparticle, and then it's intramuscularly delivered. And then there's obviously processing, translation, and communication with our immune system so that antibody production is carried on. So those are great, and those are great success stories of nanomedicine. But traditionally, there have been these barriers to achieving effective drug delivery and nanoparticle delivery. So maybe some of the barriers that you're familiar with is things like the blood-brain barrier, where the vasculature in the brain has this kind of immune privilege, and they have this endothelium and endothelial cells where they have incredibly tight junctions, sometimes 50 to 100 times tighter than other places. And they also have a lot of metabolic activity and efflux transporters where things don't necessarily get through into brain tissue. And so that can be a barrier to drug delivery for different types of neural applications. Another kind of more well-known barrier to drug delivery is the first pass effect, where if you take something orally, it's got to travel through, the GI tract, your stomach, there's enzymatic degradation, low pH, and then there's a mucosal lining, and it has to kind of absorb past the intestinal wall. And as it's trying to go into the bloodstream to circulate, it's also got to pass through the portal vein, through the liver. And here there could be up to 90% metabolism of the drug. So what's actually left in circulation is potentially 10% of the original drug dose or even less. Another barrier to nanomedicine is the reticular endothelial system, or the mononuclear phagocytic system, where our bodies kind of recognize nanomaterials as these foreign objects that are kind of on that scale of viruses. And so they'll have opsonins or antibodies that get tacked onto them, and then they get uptaken by macrophages and other types of phagocytic cells, and then they get sequestered and degraded in the liver. Nanomedicine now, we're trying to put in, and this has been done for a while, where you place things like polymers like polyethylene glycol or zwitterionic materials, so it can not absorb proteins, including antibodies, and therefore it doesn't have as much as this sequestration. And then even if you got into the right cell and it gets internalized, it might get trapped in the endosome and go down the endosomal lysosomal pathway. And so if that's the case, then your cargo and your payload might not actually get into the cytosol or to other organelles where it needs to take an effect. And so endosomal escape is something that is of high interest in nanomedicine as well. And so now there's sort of graphs like this where we're able to kind of look at nanoparticle size and charge and solubility, and we're able to kind of see some trends where, okay, if they're kind of 100 nanometers up to 220 or so, at least in this graph, they can take advantage of the enhanced permeability effect, which is this ability where there is more spaces between the endothelial cells in tumor sites, and so these nanoparticles can slip through those spaces and then reach the tumor site. Or we might say, okay, we want to make sure it's soluble versus if it's kind of hovering into the low solubility range, it might actually get more recognized by our immune system and then get sequestered. Or we might say, in terms of charge and also size, the particles that are smaller actually can take advantage of renal clearance. And renal clearance in the past has been something that in drug delivery and nanomedicine we didn't want to achieve, because that would mean that it's going to get cleared through urine. But today I'm going to talk about renal clearance and really kidney filtration as a way where we can harness and enable drug delivery to the kidneys. So kind of giving that introduction, our lab has been focused on this kind of simple platform based on these micelles and peptides for the last 5-6 years. And this is a simple particle where you have a hydrophilic group, typically peptides conjugated to fatty acid hydrophobic tail, typically very similar to our phospholipid bilayer. And depending on the parameters and some of the design of these amphiphiles, they'll self-assemble into different structures, and we design them so they become spherical. And so they're useful because they have these biodegradable and compatible components, peptides and phospholipid tails. You can allow for you can add different things like polyethylene glycol, change and tune the half-life. You can add a peptide or different agents that bind to a specific receptor or biomarker of interest, and the hope is that you can decrease off-target side effects. And you can add more than one peptide. You can add therapeutics. You can add more than one therapy. You can stuff hydrophobic small molecule drugs inside. We've put siRNA and microRNAs on the outside, imaging agents, etc. And you can make it a combination or a multifunctional system. And kind of the one unique thing about these micelles is that they can actually become quite small. Unlike liposomes or other types of organic nanoparticles that have to hover around 50 nanometers or higher, they can get down to about 6 to 7, 8 nanometers or so. And so because of that, they actually can be tailored so that they don't necessarily get uptaken and recognized by the RES system. And so they've been used for a variety of different applications in our lab. I'll touch mostly on atherosclerosis and polycystic kidney disease, although we have different types of efforts in cancer and immunotherapy and antiviral effects. And we have a growing effort in engineering extracellular vesicles, and hopefully I can share with you some of that work next time. So just to give a brief overview of atherosclerosis, this is where you have fatty plaque buildup in your arteries. Oftentimes it can progress pretty silently until a plaque ruptures. And if it ruptures in a smaller artery, blood vessel, or it's a small rupture, doesn't really cause too many problems, it can be fairly benign. But if it ruptures in a major artery like the aorta and completely obstructs a blood flow, that leads to myocardial infarction, potentially downstream a stroke, and potentially sudden death as well. So to kind of give you the cellular and molecular aspect of athero, this is a cross-section of a blood vessel. You've got blood flowing here. You have a monolayer of endothelial cells, smooth muscle cells down here. And when you have low-density lipoprotein cholesterol particles that are kind of flowing in your circulation, they actually enter into this space and they get oxidized and start to cause an inflammatory response. And so those endothelial cells become activated and they'll recruit different immune cells like monocytes. And then the monocytes, they actually squeeze into the medial layer and they start to differentiate into macrophages, and they'll start gobbling up those cholesterol particles, and they'll change into this foamy looking cell called a foam cell. And so at that point, your body starts to kind of remodel and try to really block off the inflammation that's happening here from the lumen. And so the smooth muscle cells actually, when they were dormant before, they changed their phenotype into this like proliferative migratory phenotype. And they started laying down extracellular matrix proteins, and then this is called a fibrous cap. But this area now becomes pro-apoptotic because gas and nutrient and waste exchange isn't as readily exchanged and available. And so it leads to apoptosis of these cells, and they start spilling out the lipids that they had ingested, and that leads to a plaque. And then the proteases that are also intracellular and exposed, as well as other calcification, that is happening here by smooth muscle cells that have trans-differentiated into kind of osteoblast-like cells that all contributes to potential breakage of the fibrous cap and then a blood clot. And so this is sort of the macroscopic view of how atherosclerosis and your blood vessels changes. You've got, occlusion happening and potentially a plaque rupture. But in this case, you can see that it's not completely obstructed. And so kind of earlier in my postdoc days at the University of Chicago, I started thinking about, looking at the imaging technology. And of course, there's 4 main ones that are kind of the classic imaging technologies: angiography, ultrasound, CT, and MRI. And they have different types of advantages and disadvantages to their modalities. But one thing that really struck me was this is within the same patient, and they have 2 different types of imaging. You've got CT and you've got an angiogram, and you can see here where in the larger arrows, you see that reduction in lumen diameter for both. But within the small arrows, really, the CT can pick up on the plaque that resides, whereas the angiogram, the X-ray, doesn't necessarily show that as well. And so I started thinking, well, is there a way where we can molecular kind of figure out, likelihood of plaque development and plaque rupture, really more importantly. And so, and potentially early plaque development as well. And so that got me thinking about monocytes at that particular stage. And we know that endothelial cells actually secrete this chemoattractant protein called monocyte chemoattractant protein 1, MCP1, and it binds to the CCR2 receptor on the monocyte. And so I started thinking, well, can we take the 23 amino acid sequence of MCP1 that binds the CCR2 to try to really figure out, the recruitment of monocytes at early lesions. And so I just placed that sequence onto a DSP PEG 2000 tail in this case, and then mixed it with a fluorophore. This is size 7. And so this is what the micelle looks like, and they're about 15 nanometers or so. And then in the ApoE knockout mouse, I found that they bind to early stages of atherosclerosis compared to the scrambled. And this is the aorta. But between the early and the late, we find that there's actually more binding in the late stage, and that's because monocytes accumulate over the course of atherosclerosis. And so potentially we can utilize these micelles as a way to kind of dynamically track and also understand staging of this disease. And then at USC, we added gadolinium components for MRI contrast agents just to get it more clinically relevant. And we're able to show that compared to the scramble that it can pick up on plaque components as further quantified here. And we also, we also went on, let's see. We also went on to actually add a therapeutic, a collagenase cleavable link peptide to kind of flood the proteases that are there to kind of make a theranostic system. All right, so having that as a knowledge and background for some of the things that I want to kind of get into a little bit more deeply, just when I was coming to USC, the smooth muscle cells, they were kind of ignored for a little while. And there wasn't much really known about them. And there was a big effort kind of trying to understand more endothelial cell targeting, macrophage and monocytes and things like that. But it was interesting because the smooth muscle cells, as it turns out, through a lot of lineage tracing and really sophisticated animal studies since then, that smooth muscle cells actually, they differentiate into a variety of different cell types that contribute to atherosclerosis and can actually make up to 70% of the cells that are in plaques. And so, and of course, they're in the media layer. And so we started thinking, well, can we use our MCP1 micelles to, penetrate, right, the media layer, and also maybe then deliver something for the smooth muscle cells, these underlying smooth muscle cells. And the other thing is that CCR2 is also expressed on the VSMCs down here. And okay, so this just kind of reiterates that the smooth muscle cells, they turn into this synthetic migratory type of cells, and they can trans-differentiate into calcifying osteoblast-like cells, macrophage, inflammatory cells as well. All right. And so we landed on microRNA 145 as a therapeutic because this is the microRNA that is actually mostly highly prevalent in the vasculature, and it also regulates contractile smooth muscle from differentiating. So they are responsible and for regulating the phenotypic switching of the SMCs. And so using our MCP1 micelles, we put the microRNA 145. We put the sense strain, and we have an anti-sense strain here. And we mixed it in together, and we have these mixed micelles that you can see under TEM. And just to kind of show that the mirror actually is inside, we have this migration assay, this gel migration assay, where when you have the microRNA 145 in the micelle above the critical micelle concentration, right? So the concentration in which micelles form, you can see that it does not migrate down. Whereas if it's below the CMC, it'll migrate, and this is compared to the free microRNA 145. And then this is a little bit busy, but I'll just point out a couple of things that we also treated these micelles with nuclease-containing FBS, and we're trying to understand, by placing in the micelles, can you have protection from nucleases? And you can see that, so here, this is actually a background FBS band, but you can see here, this is not treated, and it still remains basically the same intensity, and it still does not migrate. And that's compared to the untreated microRNA 145 versus the treated, where you can see that there's this loss of signal because of the degradation that's happening. And so these micelles can actually protect the microRNA cargo. And this is actually a lot of data that I've kind of just summarized into this one slide. But we've done a lot of investigative work regarding, how does it get in, how does it get out of the lysosome, and we find out that these microRNA 145 MCP1 micelles are going in mostly through the caveolate-mediated endocytosis mechanism, and that although they're kind of co-localizing with endosomes in the beginning, by 4 hours, it actually comes out via the proton sponge effect, just because they're slightly positively charged. And so that was great, because then we know that they can actually access the cytoplasm and then potentially have a therapeutic effect. And so here's kind of more of the money, right, where we say, can it actually have a therapeutic effect? We've been collaborating with Dr. Greg McGee from vascular surgery. We have an IRB collecting diseased arteries from discarded tissue during surgery. And the first thing I wanted to point out is that microRNA 145 is expressed in healthy, but is actually repressed in disease. So bringing it back in makes sense as a therapeutic strategy. And you can see that when you treat it with, and this is also further characterizing the healthy and the disease, that shows that in healthy and black, the ather-protective smooth muscle cell phenotype markers are highly expressed compared to the disease that are in gray, versus the atherogenic markers are more expressed in the disease versus the healthy. And when you treat these cells, the diseased ones, with microRNA 145 micelles, you can increase the ather-protective genes and decrease the atherogenic gene expression compared to all the other groups, including free microRNA 145, as well as the micelle that contain the microRNA 145, but without the targeting peptide. And then we went on to a couple of different in vivo models. This is the early model where we have these ApoE knockout mice, and we give a high fat diet for 2 weeks. And then at the 2-week time, we give just one injection of the microRNA 145 micelle, and then 2 weeks after that, we do some analysis. And you can see that, actually I point to the right side here, that even after 2 weeks, we have a high expression of microRNA 145. And this was a little bit surprising to me, just because in vitro, usually microRNA 145 by 3-5 days, it's pretty much gone. But in vivo, something about the pharmacokinetic properties, especially because even though the first half-life is about 3 hours, it persists. It has has a long second half-life, kind of long tail at the end, where it takes about 7 days for full clearance. And so it seems we're trying to tease this out now, but this is something that we're trying to understand, just trying to thinking about dose regimen for future clinical implementation. And then on the left side, it just shows again that we can increase our ather-protective genes, by our micelles compared to all the other groups, and also decrease the synthetic markers. This was the early stage, but we went into the late stage model where we gave a high-fat diet for 17 weeks, and we did a regimen where we gave 5 injections every 3 days. We find out that there's less plaque compared to all the other groups. That's further quantified here. And you can also see that under histology, where the microRNA 141 micelles has very little plaque, and it's pretty similar to the baseline control, which is kind of at the start of the high-fat diet, which is also further confirmed here. So there's a lot of different things that we're doing with this project. We're, right now the siRNA/microRNA clinical standards, benchmarks that we're trying to kind of mirror after has about a 3 to 4 week intravenous infusion or injection regimen. So that's something that we're trying to tease out. We're also doing a lot of work on patient background and how they respond to microRNA 145 micelles, just trying to get a better sense of and a more of a rational approach for clinical design, kind of a more rational approach for patient recruitment and clinical design. So that's kind of where we are right now. Okay, so I'm going to switch gears just a little bit here, where I'm going to talk about a small little story regarding atherosclerosis, but then transition into our kidney work. So a while ago, CRECA micelles, which is this 5-amino acid micelle that binds to fibrin of clots, was previously made into a micelle using that DSP peg tail. And in this older paper, this is actually a PNAS paper from 2019 or 2009, sorry, that showed that these micelles actually bind to atherosclerosis or atherosclerotic aortas. And I was trying to understand their pharmacokinetic properties, trying to understand, their distribution and half-life as a function of concentration effects, really trying to understand toxicity, hemocompatibility, kind of doing that extra toxicology studies. But one thing that I saw really interesting with these micelles were that at certain concentrations, they would get to the kidneys really well. That's compared to a lot of the other types of micelles and other particles out there that get into the liver via the RES system, although our micelles also can penetrate the kidneys as well. But you can see that when they're smaller, they really get skewed towards the kidney side. And as I mentioned before, in drug delivery, we kind of think about, okay, going to the kidney is not a good thing because that means that they're not in circulation and their pharmacokinetic properties don't allow for a sustained drug delivery approach. But I started looking at it and really thought this was an opportunity where maybe by utilizing size and some of our micelles, we can actually allow for, and we can design it properly where they can get it to the kidneys, and then we can design it so that it can get retained in the kidneys for a kidney disease. So if you're not too familiar with our kidneys, we've got 2 of them, and they are our main kind of blood filtering organs, and they have these individual units, filtration units called the nephron. We have about 1 million or 2 of them. And if you kind of zoom up over here, you can see that unfiltered blood goes into this structure called the glomerulus. And then, metabolites and other things can get out through urine. Sometimes there's some reabsorption that's happening. And the filtered blood then goes back into circulation. So something is happening here where, large things like red blood cells don't get out into urine, right? So there's some selectivity that's happening here, almost like a sieve, and the red RBCs and other components and other proteins, large things, are able to get back into circulation. And so it's been found that, so this is the glomerulus here, if you zoom in, that there is this 3-layered filtration barrier. And you've got endothelial cells, right, on the kind of capillary side. You've got basal membrane, which is just kind of a mesh structure of ECM proteins. And then you've got podocytes on the nephron side. And you can see that they're actually pretty porous. This endothelium in the kidney is actually, it has a fenestrated endothelium. And so it has pores that are about 70 to 90 nanometers in size. The ECM, basement membrane, has kind of a crisscross, kind of meshy structure, 2 to 8, can be even larger. And then on the podocyte extensions, there are porous slits of about 4 to 11 nanometers. And so previously, it's been found that typically inorganic nanoparticles, it's been studied with kind of gold particles or quantum dots, that they're about 8 nanometers or less. They can kind of squeeze in between these spaces and get into the kidney, and then they're typically cleared out through urine. And now we're able to show that our organic particles, even a little bit bigger, 15, can actually squeeze between and get into the kidneys, and it can actually get retained in the kidneys. And I'll show you what we've been doing regarding that work. But we had to kind of start with a disease that we're interested in. And so when I came to USC, 6 years ago, I kind of, or a little bit even before, actually before I started USC and I knew I was coming to USC, I emailed the chief of nephrology, Ken Hallows, and I said, hey, do you know anyone that might be interested? I have this interesting platform, and I'd like to start thinking about a kidney disease. And he said he was interested, and he started kind of educating me about a disease, a genetic hereditary disease called autosomal dominant polycystic kidney disease, or ADPKD. And so in ADPKD, it's actually the most common genetic kidney disease. And you can see here that your kidney starts ballooning up like almost the size of an American football versus like the size of your fist, which is kind of the size of your kidney. And what happens here is that your renal cells start to overproliferate, and it's based on typically PKD1 or PKD2 mutations. They'll start to secrete fluids, cysts will form, and it starts to change the parenchyma, and ultimately your kidney function declines. And about the age of 50 to 60, even as early as 40, half the patients will reach end-stage renal disease, and they're going to require dialysis or a kidney transplant. And when I came, actually, there was actually no FDA-approved treatments for ADPKD, which I thought was a little strange just because it affects 12 million people worldwide. And I also thought it was kind of interesting that nanomedicine hadn't really thought about kidney diseases very much. And so that's sort of where the idea struck, was our micelles are naturally going to the kidneys. This disease affects a lot of people, and we really need more efforts in kidney. And kind of a second love kind of burgeoned in my lab in terms of disease applications. Since then, there's one FDA-approved drug, tolvaptan, and it was previously approved for hypernatremia or low serum sodium, but it also has disadvantages like, it only delays end-stage renal disease 5 years after 18 years of treatment, so it's modest at best. There's a lot of nausea and high urine production, and so patients can't sleep very well throughout the night. There's a drop-off rate of 23%, and for a small subset of patients, there's liver damage, so the FDA has a mitigation strategy and is always monitoring the patients to understand if the kind of the cost-benefit analysis on kidney benefit versus liver damage. And there's other types of now drugs, including metformin, which I'll talk about, that have been kind of studied as candidates for ADPKD, but they also have disadvantages. And so the nanomedicine opportunity here is, can we enhance their pharmacokinetic properties? Can we enable higher targeting to the kidneys? And can we decrease the off-target side effects? And so just showing you that I kind of do this this every year. I actually didn't update the graph with the one that I just recently did, but it's pretty much the same, where there's a lot of nanomedicine and clinical trials for cancer and even diabetes, but there's really not a whole lot for ADPKD. And we decided on metformin because Dr. Hallows showed that when you have metformin and you treat them into these PKD mice, that you can really decrease cystic burden compared to the vehicle control. And so going back to our micelles then, we found this peptide with 2 lysines, 3 glutamic acids, that's repeating 3 times with a lysine at the end, and found that these bind to proximal tubule cells. And so we placed it onto our micelles. And one of our first efforts was just to understand that they bind to the proximal tubule cells. And this is just an in vitro assays that it shows that it's higher targeting compared to our non-targeting control, and that there is cell viability across various concentrations using an MTS assay. The one thing, though, that we started looking at was the GFB a little bit more. And within the GFB, there's actually these proteoglycans that actually make it negatively charged. And there's some size, not only size, but some charge selectivity within the GFB. And you can see here that there are more glutamic acids in our peptides. So it's actually a little bit more skewed to become negatively charged. So we started thinking about, is there some tailoring of the sequence to make them a little bit more positively charged for enabling both size as well as charge selectivity for crossing the barrier and getting into the kidneys. And this is just a partial list of our library that we made. But one of our graduate students, did a variety of different things where he started messing with the repeats of these peptides. So we've got K3, our original, but we've got 2 and 1. We started changing the polyethylene glycol linker and changing the size a little bit. We also show that we made it oppositely charged, where we have actually more lysines, right, to kind of skew it to become more positively charged. And then we have some other types of all positive or all negative. And we see actually that our the first thing I want to show here was that these micelles, all of them actually, this is the glomerulus here. This is an intravital imaging video where we injected our micelles. You can see in red that they really do pass through the glomerulus and then they go into the proximal tubules. And I can show that again, where you can see that burst of red here coming up, and then you can see a burst of red also starting to go into the tubule side. So just showing that it's in part, at least, going in through the GFB and into the kidneys. And when we design it correctly, we can really skew to the kidney targeting, or the kidneys compared to the liver. What Eve found, though, was kind of interesting was when you change the polyethylene glycol linker, this was expected that the smaller particles would get into the kidneys more compared to the larger ones. This is PEG 1000, 2000, and 5000. But the one thing that we were surprised about was that regardless of the peptide sequence, these are the K3s, the K2s, and the K1, and then this is the oppositely charged, where we made it more positively charged, and I'll just say E3, E2, E1. You can see here that it almost didn't matter. It almost seemed like it shows that with 3 repeats, regardless of the peptide sequence, had actually more targeting. And we think that's because there is almost more, they're both deuterionic, and it almost seems like there's like a deuterionic strength. Maybe there is limited protein absorption that allows for the particles to actually remain the size that was closest to when we injected it. That's a hypothesis, but this is what we observed. And so we decided to continue on with actually the K3 PEG 2000 micelle, although sometimes we also use the PEG 1000 as well. The one thing about the chronic disease, unlike other types of like cancer applications, is that it's a chronic, not an acute disease. And so you really have to think about, when you're thinking about patient implementation, a kind of a friendly way that the patient can receive this nanomedicine. And of course, in nanomedicine, we've used a lot of IV injection, but that's not necessarily the kind of most patient compliant method for a disease like polycystic kidney disease, where the patient has the disease for the rest of their life. And so we started thinking about oral delivery and how we can kind of design something where it can coat our micelle so that it can have protection from low pH and enzymatic degradation. And it can also have mucosal adhesive properties within the intestines, and for some sort of release mechanism into the blood supply. So we landed on chitosan, actually. This is kind of a summary of all the different things that we did. But we landed on these nano capsules called chitosan, or chitosan nano capsules is what we're calling them, just because we found that we're able to, they have that protection, they don't degrade in low pH, and we're able to find the right concentration, ratio with the crosslinker, the size. We've done a lot of different materials characterization, which you can further read about here from our paper from last year. But we found kind of an optimal formulation, and this is actually just showing the capsules by itself, not with the micelles. And you see that when you add free rhodamine and try to deliver it, that there's this higher area under the curve in terms of bioavailability compared to the free drug, and that there was actually a lot more mucoadhesive properties, particularly in the jejunum where there's most absorption happening. And when we just use the chitosan with metformin, you can see that there's a smaller kidney size and less cystic burden compared to the free drug. And then now, just as a quick summary of where we are, we're just about to submit this paper. We put the micelles in our chitosan oral formulation, and we delivered them into PKD mice. The first thing I want to point out was that with increasing, this is a busy slide, but I'll just give you these, point out what these red things mean, is that with increasing kind of formulation, we have just metformin labeled with a fluorophore, you have micelles with the fluorophore, and then the chitosan that has the all micelle and metformin. You can have increased accumulation in the kidney. And you can see here with our formulation that compared to the disease control, that there's a lot less cystic burden overall. So hopefully we can publish this data in the next few months or so and share with you all the different things that we've been doing for this project. The one last slide I'll leave with is that we've been using, thinking about other ways to administer drugs and nanoparticles, and so we developed this microneedle patch as well, and we added kidney targeting particles and showed that you can also increase kidney targeting, and this is just compared to a non-targeting control using our microneedle transdermal delivery system. Okay, so just with that, just want to summarize that we are using these micelles, and we're also really engineering lately extracellular vesicles. That one, we have a couple of projects on that, really interesting stuff, and really want to share with you some of that work, hopefully in the future. But we're basically, our lab is thinking about organic and biomimetic nanoparticles and how they can be used to become safe agents. And we're doing a lot of personalized medicine approaches, I mentioned, with the patient-derived arteries. And another thing I wanted to point out that's also here is that we're looking at other types of genetic diseases, like the recessive version of PKD, which primarily affects newborn babies and the pediatric population. So for us, thinking about really safe platforms is sort of a big priority of ours. Okay, so with that, just want to acknowledge the lab. Particularly, there have been wonderful past and current trainees and postdocs, colleagues that have contributed to this work. Deborah and Chris Poon worked on the microRNA work. As well as Jonathan here worked on the kidney work. And I wanted to acknowledge Ken, who is our collaborator on the nephrology side, and also our funding agencies. So with that, yeah, I'm happy to take any questions. Thank you again for allowing me to present and share my work. And thank you for accommodating, the time and everything. I know we're global at this point, so thank you again.
All right, thank you. So I guess I'll see if anyone from the audience has any questions before maybe jumping in. Oh,
Hi.
go for it, Jin.
Yeah. Oh, I first need to make the unmute everyone. Sorry. think I did. Sorry. Yeah. So thank you for your presentation. I have a few questions. Atherosclerosis. You conjugated MCP1. Is there any like the size limitation for your protein peptide or protein that you can conjugate?
Yeah, I mean, we typically have to, make sure that we typically use hydrophilic peptides, so I think that probably is, in part a limitation. And for the size constraints, usually the peptide is going to dictate part of the size, and so for the kidney application, using at least the kidney filtration mechanism, it kind of hovers around 15 to 16 amino acids. That's not to say that you can't do targeting from the vasculature side, right? Because there's vasculature that's throughout the kidneys as well. And so you can go past the endothelial cells and go through, that mechanism. And we have a mechanism, or we have a project right now trying to deliver mRNA. And that's important because the mRNA is way too big for kidney filtration. That's not going to work. And so we're using a different type of approach for that. Yeah. Mm-hmm.
For the mRNA, do you need some like mechanism to penetrate the cell?
Yeah, it's almost like a baby project right now for that one. So we're just teasing it out. But right now we've been focusing on just encapsulating the mRNA onto our particle and making sure we have peptides that can, do encapsulation or electrostatic interaction, and choosing the right, because the peptide is not just good for the electrostatic interaction, but also we're using it for targeting. So, and we're kind of mixing up different ratios as well with targeting and electrostatic interaction. So kind of at early stages for that one. Yeah. Mm-hmm.
So regarding the kidney targeting ADPKD,
Mm-hmm.
do you think that the disease might change the vasculature structure or kidney targeting because of the disease?
Yeah. Meaning like and this is for atherosclerosis, cardiovascular.
Oh, kidney.
For kidney. Yeah, it's true. I think there could be. We haven't looked at it too much. Where we're looking at, the GFB and if there's more leakiness, right? Because if there's more leakiness, similar, because atherosclerosis also has some of that leakiness as well. So we haven't looked into too much of regarding that. Yeah. Mm-hmm. But what we do see though is if you were to just inject the amphiphiles, not the micelles, we did intravital imaging, or we did injected it with the micelles, as I showed you, and then we also injected it as comparison with just micelles, or just amphiphiles, and the amphiphiles do not get to the kidneys. It has to be in the micelle form. So it seems like when it's just in molecules, it gets kind of dispersed in other places, but something about being in the micelle enables it to get to the kidney. Yeah. Mm-hmm.
Yeah. Because this is a good opportunity to talk with you. I wanted to ask because so we have the collagen binding domain that might target the tumor. You might know, but we have the 10 amino acid peptides that can deliver molecules to the tumor or inflamed tissue or disease. So I was wondering if we can somehow talk about that later.
Yeah, that sounds good. Yeah. I'm happy to talk. We should really get together and talk afterwards. Yeah. Mm-hmm. Yeah.
So if no one else has a question, I guess I'll pipe in. Apologies, I have to jump out in a couple minutes for picking up my son. But when, I guess, looking at this microRNA stuff and the effects in the ApoE mice, how well do you think that, I mean, based on the, I guess, what this microRNA is doing, how well do you think that will translate to cardiovascular disease that's set up over decades versus a rapidly forming one in the ApoE mouse and inhibiting that rapid progression?
Yeah, I haven't even thought about it like that. It's true. The way that I've been thinking about it, because the statins, right, are such a like cultural phenomenon in some ways. It's hard to break the statin barrier, it seems like, in atherosclerosis. And that has been my biggest challenge, is whenever I'm trying to convince someone, it's like, well, we got statins, we have statins, it's safe, it's statins. But if you actually look at the data of statins, it actually is not the best medication. A very high, I was just writing this up in my grant, where first of all, there's like 20% where they're not respondent or they have adverse reactions to statin. And then even upon statin or cholesterol lowering, you still have a very high chance of getting a secondary event, even after statin treatment. And so I've been thinking about our microRNA formula because, yeah, first of all, we might have to make it an oral formulation just because it's, again, a chronic disease. Second of all, if it goes through an IV infusion in a chronic disease patient, how do we make it as long as possible so that it would be just as, patient friendly? Or is there a subset of patients within those that have been given statins but are still getting cardiovascular events and are at high risk, that would actually be, the right patient for something like ours? So that's how I've been thinking about it. And so I don't know if that translates from the mouse model to the, human and things like that, but kind of just thinking about even within patients, are there a subset of patients that would actually be most beneficial for the microRNA 145 formulation? Is sort of what I'm thinking. Yeah. And maybe that has to do with them, having atherosclerosis at a different rate. Maybe that has to do with atherosclerosis and not being respondent very well to statin treatment. Yeah.
Excellent. Go ahead. Was there somebody who wanted? Maybe not. But are there any other questions from audience, or are we? All right, I think we might be about wrapped up. If you have anything else, I'm sure you'd be open to emails later. At some point, we need to find some time to chat.
Good.
But otherwise, thank you for taking the time to come visit us. It's been really great to see all the stuff that you've been doing over the past 6 years, and watching all of the stuff come to fruition. And looking forward to the stuff that should be coming out soon that I guess we've ever so slightly heard about. Yeah. Many thanks, and hope to see you again soon.
Sounds good. Thanks, everybody.